I appreciate the opportunity to address you all remotely. Wish I was there in person, but I actually had shoulder surgery about a month ago, so travel has been a bit difficult. I'm here to present on DEIMoS, or Data Extraction for Integrated Multidimensional Spectrometry. It's an open source tool for processing high-dimensional mass spec data. So to set the stage, I wanted to give a little bit of context, motivation as to why we developed DEIMoS. Generally, our goal is standards-free identification. So that is the ability to collect data from an instrument and match to reference libraries that aren't necessarily obtained from running a purified sample on the same instrument. So on our instrument side, we're currently using liquid chromatography coupled to ion mobility spectrometry, coupled to tandem mass spectrometry. But as we move forward, we're looking into using structures for lossless ion manipulation, IMS, which is a higher resolution form of traveling wave IMS. Also, cryogenic infrared spectrometry has been looking pretty promising. We're starting to build an instrument there. And it seems that software tends to be specific to a particular platform, and it becomes difficult to, say, add an additional dimension when it's something tailored to, say, LCMS. Now you have a whole new third dimension, and you almost have to start from scratch. On the property prediction side, we have two paths here. One is termed ISOCOL, the in silico chemical library engine. This is a quantum chemistry-based pipeline for computing currently CCS, although we're extending it to compute infrared spectra, excuse me. And we also have a parallel arm here called DartChem. It's a deep learning network that does essentially the same thing. So far, we've only evaluated it on collision cross-section, and we've been able to achieve a 2.5% error. I'll note that standards free from these two pieces of software is only when standards aren't available. When we do have standards, we of course use them. So asterisks here. The map from our whether standards-based or in silico predicted libraries to experimental is done with a tool called MAME, the multi-attribute matching engine, which sort of accumulates what we're terming evidence of presence. I think Gary Patty mentioned that there's often a number of features in your data that support, the presence of a single compound in different adduct forms, isotopes, et cetera. We find that leveraging all of this simultaneously can be helpful in, I guess, resulting in identification, but because it's a cumulative signal, we like to call it evidence of presence. This talk, however, is concerned only with this small blue box here, which is simply the process of taking the data off the instrument and putting it into a format that is matchable against the databases that we're putting together, whether in silico, reference-based, or just from the literature. So options here, as many of you are aware, we have vendor software. So you have these instruments, they ship with software. Unfortunately, these tend to be proprietary and black box, kind of minimally customizable to a workflow. Specifically in our case, our Agilent system did not support any deconvolution in the ion mobility separation dimension. And we want to be able to apply that to tease apart our tandem mass spec. There are a number of community offerings. Some you may have heard of: OpenMS, NZMine, MS Dial. And more recently, these have expanded to add ion mobility functionality in addition to traditional LCMS. But at the time we started developing DEIMoS, these did not support IMS. So enter our solution with a free and open source implementation. In our case, it was immediately applicable, with the caveat being as soon as we got it ready. And then for some of our sponsors that are a bit scared of not having things end-to-end under control, it's good to have for like national security space. So ultimately, we just wanted to get things from a vendor format into a place where we could process it with Python, because at that point we have a rich ecosystem of data science packages, signal processing, et cetera, that we can leverage such that we don't have to reinvent the wheel. These data aren't particularly unique. It ultimately ends up just being a high dimensional array, and there's lots of functionality exposed to be able to handle that type of data. So at a high level overview, we have our three-dimensional feature, one such extracted, visualized as best we could in 3D. Down below, we have the various two-dimensional on the top row and one-dimensional projections of this 3D representation of a feature. This is effectively the format we try to get the data into, and then we can perform these various steps on it. So input prep, peak detection, property calculation for going from, say, drift time from IMS to CCS, isotope detection, MS2 extraction and deconvolution, and then alignment, both reference-based and cross-sample. So first off, input-output, we start with typically a proprietary format, but we need to get out of that to be able to read things into Python. We did not try to implement anything ourselves in this domain, simply because ProteoWizard already has put quite a bit of effort into this to be able to leverage Agilent's inputs, Brooker's, Waters, etc., into MZML. Once it's into MZML, we then parse it into a more efficient format. We're using the hierarchical data format to get the text-based representation down in file size footprint. It also takes quite a bit to read in a 3D MZML, so with the LC, IMS, and MS dimensions simultaneously. In fact, it can take just as long to parse an MZML as it can to process the entire file, which is pretty crazy. Once we have it in a format that we can operate in Python with, we perform feature detection using a multi-dimensional convolution of a maximum filter. So that effectively is just taking our large array of data, scanning a kernel of a typical peak size in each dimension, and then we evaluate wherever the result of the maximum filter equals the original array. We flag that as a maximum, and then in addition, you can calculate other peak statistics for later down selection, because the maximum filter is pretty naive and will only give you back local maxima. Here, we're just showing an example of detecting this peak here in its three-dimensional coordinates. I will tease, though, that this approach is what we published on, but over like the last week or two, we've been exploring a topological data analysis approach, which happens to do basically the same thing, but with way less computational overhead, a much smaller memory footprint. It takes just a minute and a half on a laptop, as opposed to around 40 minutes or so, based on the convolution of the maximum filter. So that's pretty cool stuff, although we need to validate it a bit more. Drift time to CCS calibration and subsequent conversion is pretty straightforward, just following a published method by Stowe et al. From 2017. This is a single field calibration. For those of you that don't work with IMS, this probably just looks like gibberish to you, but ultimately the functionality I want to communicate is that we can do CCS calibration across different IMS types. So we've tested this on Agilent, Waters, and Brooker systems, both drift tube IMS, traveling wave IMS, and trapped IMS, given you have a representative tune mix to perform the calibration. On the isotope detection front, we kind of want to rename this functionality, honestly, because ultimately all it's doing is looking for what amounts to mass motifs in the data. So different spacing in mass can be of relevance, both in terms of isotopic signatures or even differences among adducts. So you could have, say, a protonated and associated form that are 22 Daltons apart, looking for those relationships in the data. Making that automated is makes things fairly convenient in terms of combining signatures across the many representations of a given molecule in your data set. But ultimately, I mean, you can just generalize what's shown here to adducts. But for isotopes, we have our isotopic distance is enumerated over the number of heavy substitutions times whatever the mass difference would be, so around 1 Dalton, and then divided by the charge. We can then by these putative spacings and then combine them against a candidate spacing. So we look for, say, n repeats of 1 Dalton spacings within some tolerance. And it's able to do this fairly quickly, even in the higher dimensional space. On the MS2 extraction side, and this is ultimately what caused us to want to bring our tool into our own control, was to be able to do deconvolution with IMS data. Naively, you can think of this as taking a given precursor mass. Here we're highlighting this around 663 mass to charge. You find its footprint in the drift time retention time space. The assumption is that the MS2 scan will be co-eluting and co-drifting with the precursor. You can then grab said footprint in those 2 dimensions and then report back what fragments appeared in the MS2. However, doing so naively results in some problems. First, and this ended up being actually quite a bit of effort to figure out, was that there is an offset between matched MS1 and MS2 scans, despite being just taken in an alternating fashion. So we do MS1 scan, MS2 scan, back and forth. This is actually for a tune mix of known composition, and highlighted blue are the precursor M/Z at 602-ish, and in red, the bold ones represent the unfragmented precursor and its isotopes. So you'd presume that these would have exactly the same drift time, but in reality, because of the voltage applied to the collision cell, the MS2 is accelerated relative to the MS1, and so you get this offset in drift time. This happens to be nonlinear. It's both a function of the drift time and the collision energy. We have a pretty cool way of correcting for this, and I think I have a slide on that. I can't quite remember. We'll get back to it if it is there. But here I'm showing a correction of the apices of these unfragmented precursors to where now they're aligned. But you'll notice that here in this box, I'm showing MS1. We have our mass of interest, the 602, but we also have these things in the 500 to 600 range, which would fragment alongside the 602 and contaminate, for lack of a better word, our fragmentation pattern. And highlighted in this pinkish orange color, whatever, are those corresponding to the 500 to 600 range. So what we can do is take the pairwise correlation after correcting for the MS2 offset of the MS1 and MS2, and then filter these out based on how well they correlate. Based on this matrix here, you can see that we kept the dark blue down below and filtered out the light blue, faded away into the distance, and compared against an acquisition by targeted Orbitrap. And it turns out we picked up the same fragments, albeit at slightly different intensities, but that's to be expected, as the collision energy applied wouldn't necessarily be identical, despite trying to make them as equal as possible. Oh, yep, here we go. Here's our offset calibration. So this, we were able to use an affine transform to take the projection of the data in the retention time versus drift time space. On the left, we have the difference between MS1 and MS2, a difference image in their intensities. And on the right, we have an after image, so after applying the affine transform. And you can see the intensities do reduce a bit, and you get less of, it's kind of hard to see, but there's less of a blurring across drift and retention times. So we essentially apply this matrix, which applies stretch, scale, skew, varying the affine transform, and then we minimize the loss of A multiplied by our MS2 composite and MS1. We found that the loss function that worked best here was cosine similarity. Seems to be the case in a lot of these types of applications. And you can see over here, we have the total ion mobilogram, which is an analog for IMS in drift time. And in dark red, we have after translation by the affine matrix and before in the light red. So we do get this shift, nonlinear across the drift time range, and this can be done without any calibrants. You can just use a data file that has both frames for MS1 and MS2. So pretty cool result there. On the alignment front, we can perform both reference-based alignment, which I don't know if that term necessarily means anything. It's essentially just taking some sample as given, or the reference, and you align all other samples to it based on internal standards, or maybe just putative matchings based on, for example, if you're trying to align retention time, we could find putative matchings in M/Z and drift time. What's shown here is a purposefully difficult problem where we had a degrading LC column. We didn't know it at the time, so this ended up being a cool test bed to test the limits of our approach here. And so we had the putative pairs in drift and MZ, and then plotted retention times with a pretty wide window, and then fit a radial basis function support vector regression, so basically a nonlinear robust outliers regression to the data. And then we can apply this transform to take what looked like before the total ion chromatogram. We have these major peaks here that are misaligned, and then after they come right on top of each other. This isn't a typical setting though. This is kind of the extreme case of things working with particularly bad data. Most of the time, the relationship here can be linear if it's collected, under the same conditions, and then slightly nonlinear if you're doing like intralab studies. But of course, that can vary depending on the column selected and other parameters chosen. But more importantly, we have cross-sample alignment, which is the process by which you, say, collect data over n samples, different treatments, patients.
Not to interrupt, Sean. Maybe in the interest of time, move on to the conclusions.
Okay, yeah, appreciate it. Thank you. So yeah, availability, it's on GitHub now. We have read the docs up. Most of these are filled out. A few we still have pending, so a good tutorial on how to use. You can also check it out, our publication in Analytical Chemistry, just a month or so old now. And then finally, I would like to acknowledge everyone who made this work possible, particularly Christine Chang, who presented not too long ago. If you have questions, maybe you can talk to her in person. She's pretty up on all things DEIMoS, and maybe you can have a chat at the poster session or something. Thank you for your time. And is there time for questions or just moving on?